Yeah. Okay, just turn on the medallion. Yeah. Okay, so let's start again. So it's a pleasure to have Professor Curtis Callan here with us, and he's going to be talking about the evolution of the immune system. So thank you for being here.
Okay. Is the loudspeaker working? Sounds like it is. This is good. Okay. I know that the subject of this school is mathematical evolution. I don't work in this field, and I warned the organizers about the fact. That's okay. You can talk about whatever you want. So I will talk about things that I have been doing over the past number of years. See, now it stops. What's going on? Why is that? Reception, I guess it must be. So we want to make sure that the...
Put it a little off. You can put it a little farther. It has a bit more of a peak up, if you don't mind.
Okay, no, go ahead. Put it. Let's make it work. It's the cutout that's the problem.
Let's see if it works up better.
All right.
Like this. Try it.
Okay. And now? All right. I will try to talk firmly and authoritatively, and maybe I will be heard anyway. I think it matters whether I do this with my head or not. Okay. Where is the radio? Over here. So if I'm nearer here, it's better? Okay. Well, that actually works. Okay. Very good. I do tend to pace. Okay. So I'm a theoretical physicist. During most of my working career, to distinguish from my current career, which is not a working career, I mostly did particle theory, and I got interested in biology, and I got drawn into corners of biology that basically have to do with cellular biology. So we're not going to the school, I guess we are not going to be in the realm of cellular biology per se. We'll be more in the realm of biology of populations and evolution. So what I would like to do is to talk about a couple of aspects of cellular biology that connect with things that are quantitative and semi-mathematical, and I hope you will find it, so to say, culturally enhancing. If not, you can go to sleep. All right, so this is just a sampling of the kind of papers, of the kinds of projects that I've worked on in the past 5 years or so. And as was said by Marcos, what I really want to talk about in the end is work that I've been doing on the immune system. But since I have 3 lectures or so to talk in, I think you'd get sort of bored if I talked about mathematical immunology for that many lectures. So I thought I would do a kind of potpourri of things that come from these other subjects. It all, one way or another, has to do with how can you make sense of what cells are doing in a quantitative way. I mean, biologists are extraordinarily good at observing and observing what cells do. Physicists want to construct mathematical frameworks in which you can understand in some quantitative way, basically writing down equations when it comes down to it, how cells behave. And that's kind of the framework within which I am going to be working. So what I will do to start with is describe a project that the first graduate student that I worked with when I became interested in theoretical biology a la modern times. And it's, the title is Deep Sequencing, Mutual Information, and the Thermodynamics of Gene Regulation. So, as we all know, have a genome. This genome is very capacious, even in as simple a thing as an E. coli bacterium. The genome contains the code for something like 5,000 different genes. And these genes are not all expressed simultaneously. That would cause a traffic jam of immense proportions. Basically, some few hundred of them are indeed expressed all the time because they deal with functions of the cell that are always on, but a lot of other things are expressed only when needed. And I guess at some point it was mentioned the Lenski experiments in which the cells were made to eat arabinose, which is a rather bizarre sugar. They really like to eat glucose, and they have to learn an apparatus that can turn arabinose into whatever elements the cell really needs in order to live. So there's a basic question of how cells regulate the expression of their genome. And much is known about this, and there's a basic process in which, well, the genes themselves are coding proteins. There are certain proteins whose whole role in life is to turn on and off the transcription of particular genes. And how does that work? Well, so we have up here a notional stretch of DNA, and starting here, there's a sequence of bases which code some useful gene. And there's something called a transcription factor, which is a protein that has been made by some other gene that, because of some change in circumstances, has taken on a configuration that makes it possible for that protein to be attracted to a particular short sequence of DNA that is upstream of the coding region. And when it sits down on that particular stretch of DNA called the promoter, it enhances the likelihood that the big enzyme called RNA polymerase, whose job it is to, eat molecules and simply this is the problem with this thing, and transcribe the coding region into RNA, which eventually into protein. There's an interesting problem in that what makes this little stretch of DNA that's not coding for anything, what makes it a promoter, is a particular structure of sequence of DNA. In other words, there's a pattern that makes it attractive to this transcription factor and not some other transcription factors whose mission in life it is to turn on and off some other gene. Okay, so let's. So there's a coding problem. In point of fact, there are many transcription factors that actually regulate multiple genes at the same time, and you discover that in point of fact, these sequences that are attractive to that transcription factor are not all identical. In the end, we believe that this is an energy problem. That is to say, these transcription factor proteins have the feature that they have some element of their structure that is designed to essentially recognize a particular DNA sequence. Why not? Proteins are extremely versatile, and there's a class of protein structures that are made to sit down on the DNA and basically give you a particularly strong attraction between protein and DNA when the right sequence is sitting underneath it. So there's an energy, so you can cast this as an energy problem. That is to say, transcription factors maybe can be said to be generically attracted to DNA, but they're strongly attracted to particular sequences of DNA, and it would be very nice if you could get a, how shall I put it, a dictionary that says DNA sequences that have this structure are attractive to this kind of transcription factor, and eventually have some kind of mechanistic understanding of how it is that gene expression is regulated. Now, so there is an energy problem, and the problem that we set ourselves initially was how to go about in the best possible way of finding where these promoters lie, that is to say, where on the genome of your species of interest these promoters might lie, and how can you characterize the binding energy between a transcription factor and some promoter sequence that's possibly not exactly what you have decided is the best possible sequence. After all, things do get mutated. Okay, so you can cast problem into something that is close to an energetic problem. You'd like to be able to get access to the in vivo energetics of gene regulation. And so this student with whom I worked some years ago now dreamt up a really rather interesting experiment for addressing this question. So the framework was to try to get quantitative insight into how do you understand the binding energy of a particular transcription factor to DNA sequence, and how do you identify the DNA that it is most attracted to. And so the idea was to adapt high throughput sequencing and the methods of molecular biology to create a data set that could then be analyzed in hopefully intelligent and innovative way in order to get the kind of answer that we're looking for, namely some kind of mechanistic model for the energetics of transcription factor binding to DNA sequence. Okay, roughly speaking, well, I have some ideology here, but I think the ideology is probably best explained in context of what we actually do. Okay, so there's an experiment to be done, and this is an experiment that I would say the biologists would not have wanted to do by themselves. It's kind of a physics molecular biology experiment, and the student was willing to, or he was eager to learn the techniques of molecular biology to be able to carry out the experiment, and there was a friendly molecular biologist at Princeton named Ted Cox who thought it was rather cute to see theoretical physicists messing around in the lab, and he made his lab available. And, you know, so what was done was the following, all right? So the thought was that, okay, so the actual context was the so-called Z motor. So E. coli is a bacterium that eats sugars. It really loves to eat glucose, but if glucose is not available and lactose is, it will eat lactose. But in order to eat lactose, it needs things. It needs a surface that basically allows the lactose molecule to get sucked into the inside of the cell, and it also needs an enzyme that can do the conversion of the lactose sugar into some more primitive energy-containing molecule. You think it's better for me to use that?
The computer person thinks it's the reception of that microphone.
All right, do you think this will? Okay, I have to turn this off so I don't squeal. All right, does this do any better? Okay, all right. Okay, well, I'm not sure I can talk and hold a microphone and a pointer at the same time, but I shall try. All right, so the thought, okay, so there is a particular gene and promoter which is essential to the E. coli bacterium in order for it to eat a special sugar, lactose. And for various reasons, that region of the E. coli genome has been, not just the genome, but the functioning of that group of genes has been studied to a fairly well, and an awful lot is known about it. So our thought was to do the following set of experimental manipulations. All right, so here is a model for the thing that we're about to do. So in the organism itself, there's a gene. Actually, there's 3 genes. There's LAC-Z, LAC-I, and LAC-Y, I think. They're all transcribed at once they're transcribed, and there's a promoter region upstream of that group of genes, and we'll just call it the LAC-Z promoter. Now, the idea was, there is some, you know, if you go to the genome database and you look at the genome of E. coli, which by the time we were doing this, of course, had been sequenced many times over, you will see that, well, in the standard laboratory strain of E. coli, there is a particular thing that's known to be the regulatory sequence. And our thought was, okay, let's on purpose induce mutations, that is to say, modify that promoter in some semi-random way, and ask, well, what do these mutations do to the actual functioning of this promoter? So instead of actually having the promoter cause the genes that are useful for metabolizing LAC-Z to be transcribed, we instead wanted to induce the transcription of a so-called fluorescent reporter. That is to say, it's a gene which, under the appropriate optical stimulation, will glow, all right? And so by the time when you've put something like this into your cell, if you stimulate it with the right kind of laser light, it will glow, and the intensity with which it will glow will tell you, so to say, with what efficacy, with what efficiency, oh, I'm sorry, I'm doing what I'm not supposed to do. So to say, the different variants of this sequence that have modified the promoter region will be in any given cell, so to say, translated into a different level of fluorescence. Okay, so that sounds pretty simple. So what you do, basically, is you make use of the technology of molecular biology to create a so-called plasmid library in which you put together the promoter that you're, how should I put it, the promoter region that you're interested in, the fluorescent reporter gene, and a bunch of other things, basically an antibiotic resistance gene, which will allow you to pick out those cells that have actually taken up this little circular fragment of DNA, because only they will survive attack by some antibiotic. And with the help of DNA technology, you can essentially create randomly varied stretches of sequence which are going to represent the mutations of the promoter region, and all the rest of it can be put together from standard sources. And so what you do is you create what's called a reporter construct library, that is to say millions and millions of different variants of the promoter together with, on a circular bit of DNA, the other things that are needed to make this useful. Then you, so to say, transfect good old E. coli with these guys. You subject these E. coli bacteria to some kind of shock. Electroshock actually works pretty well, apparently. And many of them, some of them will take up one or more, and you try to control things so that at most they only take up one of these little circles. And then you let them grow up, and then you basically run them through a cell sorter, and you select, you divide them into batches according to how brightly they glow, all right? So basically, everybody that's taken up a promoter sequence variant that is really good, is really strong at producing the fluorescent protein, they will glow strongly. Those that have taken up variants that are really bad will glow less strongly. And basically, you run you grow the E. coli up in a pot, you run them through a so-called cell sorting machine, and as they are going through the cell sorting machine, you basically sort them by, you divide them up into some number of bins according to their fluorescence. So the output is a certain number of bins which contain a lot of sequences that have different levels of fluorescence. And with the wonders of modern sequencing technology, basically you can obtain the sequences of these varied promoters. And the way you do it essentially is, well, there's a whole technology of PCR and the rest of that, which I won't go into, but basically in the process of carrying out the sequencing of this DNA, you add little barcode tags to the individual varied promoters, so that when you get the sequences back from the sequencing company, or from whoever has a sequencing machine that they're willing to let you use, you will have not only the sequence of the varied promoter region, but you'll also have a tag that tells you, did it come from the really bright group, did it come from the medium group, where was it in the general range of fluorescence. So this was done 10 years ago, and the number of reads that we got was not the maximum we could possibly have gotten, but we got out something like a couple of 100,000 reads in total. We did several different experiments. What did we vary? Well, we varied the mutation rate within the promoter region. We varied where the mutations were done. For example, it turns out the whole region of interest, the region that contains the stretch of sequence where the RNA polymerase binds and the stretch of sequence where the transcription factor binds is about 75 base pairs. So we did in some cases, we basically randomly mutated at a 12% rate within that whole region. In a couple of other cases, we varied either within what was known to be the binding region of the transcription factor, but not at the polymerase binding region, and vice versa. And we also did experiments with an engineered strain of E. coli that had been made by a previous postdoc in our biological collaborators' lab, where we could exogenously control the signal that made the transcription factor want to bind to the DNA. In other words, how does a transcription factor know that it's time for it to do its job of binding to the DNA and enhancing or not, as the case may be, transcription of its gene? Well, there has to be some chemical signal. And so there's a chemical signal called CRP, and when that small molecule, let's see, for some reason, I can't tell you the exact name of the small. This small molecule becomes, its concentration in the cell goes up when glucose is not available, and when its concentration goes up, it interacts with the protein that eventually is going to bind to this regulatory sequence, and in effect makes it capable of binding to the DNA. I might go into that a little bit later. At any rate, controlling the amount of CRP within the cell is something which the cell would like to take care of on its own, but if you disable its control mechanisms in a strain of E. coli, you can essentially control how much CRP there is in the cell. So we also did experiments in which we varied the concentration of this chemical signal within the cell. All right, so that is the experiment. And why did we do this, and what was the outcome? All right, so here we have just a summary of what the data looked like. We had tens of thousands of instances from these different, tens of thousands of instances from each of the experiments. Each of the experiment divided these instances into some number of bins. And so, for example, this leading set of experiments, so to say, in each bin we would have on the order of 5,000 sequences. We thought that, for us, that was an impressive number. These days, one multiplies all these numbers by factors of 10 or 100 if you're so inclined. But the real point eventually is going to be, this was more than enough to actually extract interesting physics. Yeah?
Do you have any control on whether the transcription factor that binds is always the same?
Yes, I would say yes. So there is certainly an interesting issue. Transcription factors have typically a generic affinity for the DNA molecule. The DNA molecule at one level is just a random string of bases, and if you have 6 million or so randomly assorted bases, maybe every now and then you'll get a sequence that is attractive to somebody's transcription factor. So there's an interesting issue there, which maybe I'll come to when I talk about tomorrow. Okay, so let's soldier on here. And okay, so here's a cartoon which is worth remembering. So in the cartoon you see this is the stretch of DNA that we mess with. This is the polymerase, and it likes to bind, and this arrow indicates the start of transcription of the gene. This is a map of the promoter of the plasmid that we constructed in order to play this game. And here, for example, is, this is a plot of fluorescence of cells as they're fed through the cell sorting machine, and you can see the distribution of fluorescence, and here are, a set of bins that describe how we pick things apart. Okay, and here's a more complicated map of the plasmid, or at least the business end of the plasmid. All right, now, okay, so what was the idea here? What was the first thing that we did with this? Well, the first thing that we wanted to know, let's see if this is the right place to go. Let's go here. What did we want to know? Well, we wanted to know, are there particular places within this window that are important and other places that are not important? So that is to say, important in the sense of having an impact on how well this promoter functioned as measured by how strongly this fluorescent protein was made, the fluorescent protein substituting for the gene cassette that was useful for metabolizing lactose. Okay, so basically we asked, take a particular position here, like let's take minus 60 or something like that. In position minus 60 in the reads that you get out of your sequencing in a given experiment, you can have 4 possible outcomes, A, C, T, or G, 4 bases. And you can ask, what is the correlation? Is there any correlation between the base that you see in that position and the level of fluorescence? That is to say, how well the promoter is actually functioning. And we measured that basically using mutual information. So on the one hand, we have tens of thousands of examples. For each of the examples, you have it fell in such and such a bin in the fluorescence measurement, and at the same time, the base of that position is A, C, T, or G. So you have a joint distribution between where it comes out in the binning of fluorescence and what is the base of that position. So you have a joint probability, P of B and mu, where mu indicates the bin for the fluorescence and B indicates the base at that position. And so there's a standard construct when you have a joint probability distribution between two variables to know in a principled measure of the correlation between the two of them. How many different levels of significance can you get out of one of the variables by measuring the other one? Yeah.
This is your entropy.
Sorry?
It would be something like an entropy.
It is not unrelated to entropy. Yeah, but it's literally this. So the notion of mutual information between two correlated variables was introduced by Shannon in 1948. He asked for some general features like, well, if they're uncorrelated, it should be 0, and stuff like it should, in a certain sense, be additive, et cetera, et cetera, et cetera. And the formulation that is extremely useful is as indicated here. That is to say, it's the log of the joint probability divided by the P of B and P of mu are the, so to say, how do we call it? P of B is the probability of finding the base independent of mu, so I've marginalized over one of the variables. P of mu is what I get when I marginalize over the base, and I take the ratio of the joint to the product of the two marginals, take the log base 2, and take the average of that over my entire data set. It's in bits, so if the answer is 1, in some sense that's 1 bit. That's 1 factor of 2. That says that, roughly speaking, when you look at what you get for one of the variables, you can tell basically whether or not the other variable lies within, so to say, 2 different bins, high or low, Right? It need not be high versus low. It might be two categories into which things are distributed in this way. Don't have to be, it's not a linear regression sort of thing. Okay, so what one calculates this quantity for all of the bases, all of the positions that lie within this varied promoter region, and you ask, well, is the answer 0 or non-zero? Okay, so the answer is non-zero, and the non-zeroness is geographically extremely informative. That is to say, you would expect that in places where neither the polymerase nor the transcription factor are interacting with the DNA, you would expect that the answer wouldn't care much about that base. And those are places within this map, if you will, where the value of the mutual information is small. In places where it should matter, because you know or you suspect that the protein that's touching the DNA is touching the DNA there and is somehow sensitive to what are the values of the bases, you would expect that there should be some meaningful correlation between the value of the base and the level of transcription. So this is what we call an information footprint. And in this case, we knew from previous information what we should find, but okay, this is an indication that you are indeed finding the right thing. That is to say, this transcription factor, called CRP, it's a dimer, and it turns into a dimer when the chemical signal is present, and the dimer basically has 2 feet that touch down on the DNA, and you see the 2 feet, you see a nice symmetric pattern. Also, it's known that the polymerase has 2 major places where it contacts the DNA, and you see the values, the non-zero values of mutual information there. So the point values of mutual information are small, but they're very significant. And, we showed here what are the 1 sigma error bars. The lower curves are blowing up with the bigger curves so that you can see the actual values, even though they're in tiny fractions of a bit, they're meaningful. Okay, so this is to tell us that we're not doing something that's totally crazy. And now, okay, and things look different as you do the experiment differently. In the cases where you get to control, maybe I will just point to 2 things. So in the cases where you can vary the intensity of the chemical signal inside the cell, you see that when the signal is strong, you reproduce the previous result. As you decrease the strength of the signal, what's happening is that you would expect the importance of the transcription factor binding to go away, and indeed it does. Okay? So you can learn something significant on a functional basis from looking at these things. Now, all right, so that's all very qualitative. It's just saying by doing an experiment of this kind and using mutual information to ask, well, what positions are significant, and quantitatively how significant are they? You can verify things that you already know, and you can also learn things that you didn't know. Now, what we would like to do with this is a little bit more sophisticated. So the other side of the coin was, I started by saying that we believe this transcription factor binding is an energetic issue, and that at the heart of it all is, for a given kind of transcription factor, there is a function that basically takes in the DNA sequence, so to say, underneath the place where the transcription factor sits down, and tells you, as you vary the choice of bases at different positions within this window, tells you how the binding energy varies. All right, and so what we're seeing is that, well, we haven't seen the details of that, but that's what we would like to figure out. Now, our data set is a big collection of pairs of sequence of the promoter region and observation of level of fluorescence or level of transcription of the gene that's driven by this promoter. We would like to have a model for how the binding energy of the transcription factor depends upon the sequence that's in the window within which the transcription factor sits down. Okay, if we had such a model, we could convert our data set to pairs in which we have the observation, the sequence, the fluorescence level, and the energy, the binding energy of that sequence. So mutual information between these predictions of the binding energy and the observation of efficacy of transcription would be an unbiased measure of how well the model predicts the data. Well, all models that you can imagine have parameters, right? So, and we'll come to a specific modeling strategy in just a second. And basically, what I'm leading up to is that what we'd like to do is find a way to use the data that we've generated to essentially decide within the framework of, I have some parametric model for how the binding energy of the transcription factor depends upon the DNA sequence in its window. How do I figure out what's the best choice for those parameters? Well, what I want to do is, this time I'm going to say, I want to find the set of parameters that maximize the mutual information between the thing that I observed, which level of fluorescence intensity, and this thing that I'm predicting, the predicted binding energy as a function of sequence. That depends on parameters, and I just have to explore a dauntingly large parameter space to find the best parameters that maximize the mutual information between prediction and observation. Okay, and basically there is a procedure. Basically, we used some variant of the classic Monte Carlo method to, in effect, find what are the best parameters. But first, before I do that, I have to tell you a little bit more about how we parameterize the model. Okay, so a typical parametric model, this is something which people have been using and understanding, or so to say, organizing information about transcription factor binding for years and years and years now. Basically, you imagine that you have some footprint on the DNA, that is where the protein of interest is actually touching the DNA, and you, at each position, you get to make a choice of one of the four bases. We can interpret this, let's see if I say this here, right. We can interpret this as a machine for calculating the binding energy. That is to say, if I put numbers, if I put numbers into each of these boxes, so in position 1, the first position, if I choose A, I put in one number. If I choose C, I put in a different number, G, T, etc. Basically, and essentially make the assumption that the binding energy of this transcription factor to the sequence of DNA is basically just the sum of the numerical contributions from each column. And so in each column, in reality, there is a particular choice. We will attribute each particular choice at each position as contributing a certain amount to the binding energy, actually the negative of the binding energy. Big is bad here. And in principle, if we want to know what is the binding energy to an arbitrary sequence, you have to fill in all the numbers in this table. All right? And the typical length of the binding of the footprint, if you like, of the CRP transcription factor on the bacterial DNA is more than 20 base pairs, so one has quite a large set of numbers to somehow wrinkle out from the data. Okay, so on the other hand, we have a procedure for doing it. That is to say, we simply take our data, We explore parameter space, which is a large dimensional parameter space, but that doesn't phase modern computers, and one can find the set of parameters that maximize the mutual information between the prediction and the observation of fluorescence. And so this is turning those energy matrices into a little mosaic where you see the various positions, you see 4 colors corresponding to the 4 different bases of each position, and somewhere there is a color code that says blue is you put a small value there, red means you put a large value there, and intermediate colors, intermediate values. So what you see is if you have a block of blue, that's telling you it really doesn't matter what choice is made for the bases in that region, because whatever you choose, you don't change the binding energy much. In places where you have one red and some softer colors, that means the base that at that position, the base corresponding to the red color is highly disfavored because it costs you a lot of energy to put it there, and the others are less disfavored, etc. Now, of course, what you immediately have to worry about is that you're getting an answer, but is the answer worth anything? This is we're supposed to be extracting a property of this transcription factor protein from your data. This is something which is meant to be universal. That is to say, it should not depend upon the experimental conditions under which you carried out the collection of the data. And so that's why I have so many different answers here. That is to say, this is clearly there is a high degree of consistency between what I'm getting for this energy matrix in these different experiments that I mentioned. And what's expressed here is the level of correlation between the matrix elements that are actually sitting in those boxes as measured in different experiments. What is the correlation between different experiments? As you can see, it's the scale here is from 0.9 to 1. So between experiments, the correlation is extremely high. So that's an indication that, well, whatever we're doing here is actually measuring something that is close to universal. We're in fact doing something that is giving us an answer for a parametrized measure of what's the binding energy of the transcription factor to a particular sequence, allowing you to change the sequence. And we're getting essentially the same answer no matter which of these various experiments we did. So it's a restricted model. It's not the most general possible model of how the energy could depend upon sequence, but clearly it captures a large fraction of reality. Okay, now, I want to go one step further. I mentioned the word energy. I should also mention the word time. This looks like 5:45, is that correct? Okay, so I think I'm doing okay. I mentioned the word energy, and I said I was constructing for you a parametric model for what's the binding energy of this transcription factor protein to a particular promoter sequence. And I was putting numbers into that matrix, but the numbers didn't have any dimensions. And indeed they didn't, okay? Because there was no way, given the way I asked the question, for me to attribute, kcals per mole or ohms or electron volts or whatever to the numbers that I was getting. So how do you do that? That would be rather interesting. So that's what this, so to say, second version of the problem is meant to do. So this is, only a physicist would want to do this, I think. All right, chemists too, I think. All right, so we can use more complex models that take into account the cooperative nature of regulation to further compress the sequences into some useful information. So there are various reasons why we want to do this, but certainly a really fundamental reason for wanting to do this is you really would like to understand whether this measurement of just pure numbers, what's the value of, well, basically measurement of pure numbers, can we in fact turn that into something that has energy units? So to the extent that we're just talking about equilibrium thermodynamics, it's possible to cast the problem in slightly different terms. Namely, let's see whether the next, okay, so let's go back here, or rather let's go forward to this briefly. I've talked so far about trying to figure out what is this parametric matrix that tells you something about the energy of interaction of the transcription factor with its bit of DNA and the energy of interaction of the polymerase with its bit of DNA. And I said, well, we extracted some structure that was consistent between different experiments. Now let's look a little bit more closely at what's going on from a thermodynamic point of view. What is the point of having this transcription factor? Well, in this particular case, the point of having the transcription factor bind to the DNA is to enhance the rate at which the downstream gene is transcribed, and in order for that to happen, you have to enhance the probability that the RNA polymerase will stick to the DNA. Supposing that it's all energetic, the only way that can happen is if there is some kind of cooperative interaction between the polymerase and the transcription factor. So there's one further element that we haven't talked about so far, which is, let's suppose that there's some mechanistic energetic interaction between these two proteins that are playing in this particular game. So there's another number, namely the energy of interaction between these two guys, that must be important. There's also, of course, the energy of interaction of the individual proteins with their stretch of DNA. So we'll back up in just a second. Another way in which transcription factors can play is basically to turn off transcription of a gene. And a really effective way for them to do that is to decide not to bind here, where with the help of an energetic interaction they can enhance the likelihood that the polymerase is going to do its thing, but simply just sit down either here or here, somehow just plain interfere with the ability of the polymerase to do its thing. That actually happens in one of the viral systems that I expect will be talked about soon, we'll come back. We'll see whether some other lecturer gets there. Okay, so if I want to think about this in energetic terms, then there's a simple statistical physics model. Namely, I have two players. I've got player A and player B, and they both have their own probability of sitting down. Well, how shall I put it? There are four states the system can have. Nobody can be bound, neither the transcription factor nor the polymerase, or one of them can be bound and not the other, or they both can be bound. That's basically all you've got. There are those three.
That's EI, right?
Yeah. Now, okay, so if you wanted to build yourself a little thermodynamic model of this, you would say, aha, well, so the transcription factor and the polymerase are in solution at some concentration. The probability that they are bound is going to be proportional to their concentration with a Boltzmann factor that tells you about their probability of being found bound. So if you write yourself a thermodynamic model, so what are the four states? Nobody's bound. Let's call that probability 1. The transcription factor is bound, but the other guy isn't, so that will only care about the concentration in the solution or in the cytosol of the transcription factor, and there'll be a Boltzmann factor that depends upon its energy, or the RNA polymerase is bound, and it depends on concentration and energy, or both of them are bound. Okay? And the only thing that's new is that both of them are bound, and there's the possibility of a contribution, a Boltzmann factor contribution from this hypothetical interaction energy between the two of them. Okay, so let's keep the same model as before for these energy functions, the things that depend upon sequence. This interaction energy is just a number, and so basically we have the same set of parameters as before with a couple of added numbers, namely the interaction energy and the concentrations in solution. So let's play the following game. This is the probability that the site that could be occupied by the polymerase is in fact occupied by the polymerase. If it is occupied, then you would say that's a good measure of the rate at which the probability of occupation of the polymerase is a good measure, let's say, of the probability or the rate at which the gene is transcribed. So what we would like to do now is to maximize the mutual information between our prediction for this occupation probability of the polymerase and our measurements, okay? And, okay, so for every measurement then is a sequence and a level of fluorescence. So there's a mu and there is a sequence sigma, and we have this model that depends on parameters that turns sigma the sequence into a tau probability of the polymerase being occupied. Okay, problem, maximize this mutual information over all the parameters, and the non-linearity of the dependence of tau on the energies, the fact that all of the energies appear as energy divided by kT, or RT, depending on how you like to write it, means that the answers are now going to be determined in actual physical units, which is kind of cool. And so it turns out this is just as doable as the other one was. And what do you get? Well, you get basically the same result as before for the energy matrices, except that the numbers that go into the boxes are now in units of energy, kcal per mole, or EV, or whatever you prefer. And in addition, a number for this interaction energy between the transcription factor and the polymerase comes out, and it turns out to be, some number, minus 3.3 kcals per mole. You also get something for the concentration of the CRP in solution, and for technical reasons, you don't get any number for the concentration of the polymerase molecule in solution. So this is just to say that the method works, and it actually gives you numbers, and the numbers are consistent. And let's go one step further. And we were kind of happy about the fact that the people who had studied the thermodynamics of the regulation of the LAC promoter, doing very careful and clever molecular biology measurements, have independently determined that this interaction energy between CRP and polymerase was minus 3.4 kcals per mole. The fact that we come this close in this is, I'm sure, a bit fortuitous, but at least it's well within reasonable limits. Okay, what else to say here? There are multiple data sets, so you can either perform your optimization on one of the data sets, or you can do it on all of the data sets independently, or you can somehow put all of the data sets together, and I think I won't go into that right now. And one of the things that I think is perhaps worth mentioning is that one of the things of interest is how well do you determine these parameters? Do you in fact, from this approach, acquire some idea of the uncertainty in the estimation of the parameters? And you do, and because the number of data points is very large, these uncertainties are actually quite modest. And I think the last thing I want to say is here. Okay. I'm looking for a particular datum here. Okay, let's focus down here. Okay, what I finally described for you was what was the mutual information per base, as it were. So what is the total mutual information between the binding probability, this quantity tau, and the observed fluorescence? So when I actually do the optimization, I get a number. I get 0.732. The interesting thing is that this is, you might ask, is this a good number, bad number, mediocre number? We also figured out what was the mutual information between just the sequences themselves and the fluorescence, and roughly speaking, that's roughly 1 bit. And so we're getting about 80% or something like that of the total available mutual information from this procedure. So this is probably not something I can pursue very far here, but we asked a variety of questions about, did you really need to put together everything in the thermodynamic model? How well did you do if you just measured the energy of the unnormalized energies of the binding of the transcription factor and the polymerase? Actually, it turns out you do almost as well. On the other hand, if you just concerned yourself with maximizing the mutual information between the paired energies and the data, you don't learn anything about the interaction energy, and you don't learn anything about the energy scale. So clearly doing this thermodynamic model is better. And you clearly also asked, well, if you simply agglomerated the energies, just took the sum of the binding energy of the polymerase and the binding energy of the transcription factor as your measure, how much mutual information do you capture? Well, significantly less than if you do the full thermodynamic model, or allow yourself to talk about the pairs, the two energies. Okay, so where am I going to go on this? I think I'm done for the day. So the experimental assay is pretty straightforward. If you can create many billion genomes and assay their performance, information footprints reveal where the functional protein DNA binding sites are located. Mutual information analysis gives you pretty precise quantitative in vivo biophysics from what is really very noisy qualitative data. And in principle, the assay can be applied to any regulatory sequence in any cell type. And we say the problem may be one of getting a large enough number of instances. I think that's actually not typically the problem. The technique looks pretty general. It may be applicable to many different problems in molecular biology, both in vivo and in vitro. And as usual, I would say we have been a prophet crying in the wilderness and not even having honor in our own country, in the sense that the graduate student, Justin Kimmey, has applied this to some other interesting variants of this problem, actually something that has to do with the immune system. I may have something to say about that coming up. And Ted Cox is the friendly molecular biologist that allowed Justin to learn how to do molecular biology as well as theoretical physics. And there was another graduate student who was involved in the analysis, but who came to fruition when we started to work on the adaptive immune system, which I will talk about. Okay. So tomorrow, tomorrow I plan. Let's see whether the plan survives contact with reality. I plan to talk about making use of these models of sequence-dependent energy of binding of transcription factors to DNA and apply them to a problem in evolution. Right? So I will read in a little bit of evolution, so I will make this a little bit relevant to the subject of the course. And because there's an interesting, really quite restricted evolution problem, which has to do with the fact that, well, if you took a look at two related species, and you look at a particular regulatory sequence, but you will discover that, well, they're not typically the same, right? If there's enough divergence in time between the species. And you can start asking, well, what is it that is selected for? Is it the sequence that's being selected, or is it the binding energy of the transcription factor that's being selected? And that turns out to have the two options have very different consequences for certain data that you can readily get your hands on. So I think it's a nice exercise, and what I did today basically was to show you how, without doing, well, it's not that the experiments are easy, it's that they're a different kind of experiment than what you normally think about when you say, I would like to measure the binding affinity of some protein to some wide range of DNA sequences. You don't actually directly measure in some mechanistic sense. You sort of say, create a set of data that is influenced by that binding energy, and using inference technology, figure out what must have been the energy dependence on the sequence. And so I want to use that in a little digression onto evolution, which I hope you will find amusing. At least it does connect with the subject of the school. And then I will pass on to talking about the immune system, which, again, it's not clear what it has to do with evolution, although it evolves. And I will be able to show you a couple of things where it's clear that evolution has done something interesting, but I have no idea how it happened. So it's an open problem. Okay, so I'll stop there. Sorry?
To have the break, or just...
I can live without, I mean, I don't know about other people.
So what about not taking a 10 minutes break? Just go on with the questions right now. We had a big break before, yeah.
Okay.
So if it's okay with you. Sure, yeah. So, okay, question there.
Thank you, Professor. So, yeah, from this framework, it seems like the probability of attaching to a specific part is quite fixed. What is your take on how would the cell, what would the cell vary if it's put into a situation where its promoter or its gene needs to be transloaded, or a certain protein needs to be made a lot? Is that so? It seems like the probability of falling into a specific place is fixed, right?
Yeah. So what I was not talking about was the competition between the place where transcription. Sorry. I didn't talk about a related problem, and it's part of the whole problem, understanding how the system works. So there's a lot of genome out there, and there's one particular location on the genome where, let's say, the CRP transcription factor can bind and do something useful. There are a lot of other places where it will have some probability of binding, and there's a significant question. If you add up, so to say, the probabilities of binding to all of the other places where it could bind, how does that compare with the probability of binding to the place where it's supposed to bind? Is that, roughly speaking?
So let me tell you. So since the genome is fixed, if you have a cell and you can make a lot of certain protein, how would you go taking into account that those energies are fixed and like the probabilities also?
I'm not getting it quite. I mean, so one of the things that we certainly get from this is if you alter the sequence of the intended binding site, if you modify the sequence of the intended binding site, well, you can say something about how does that affect the probability of binding and therefore the probability of initiating transcription. And that was the purpose of the exercise. Maybe we should try it.
Yes, actually. Thank you.
Oh, thank you. Which Monte Carlo method are you using? Which Monte Carlo method are you using?
Markov chain Monte Carlo.
Like Metropolis for your detail balance. What gives you the equilibrium?
Let's see, so you're asking me to recall exactly how we did this.
Oh, sorry.
Well,
Because I was just...
one has a Hamiltonian, as it were. That is to say, we're attempting to maximize, well, let's see, how do we do this?
Because you're trying to minimize the energy, right?
No.
Maximize the neutrality.
No, also, we're maximizing the mutual information.
Okay.
Okay, so we have to, so that's the quantity that's being maximized, the mutual information averaged over the entire data set, right, of prediction versus observation.
Okay.
Maybe we should do this offline as well.
Okay.
The lecture, there was a slide called What, Why, and Goals, and then wrote the topics, geometric considerations. How would you consider geometry on your experiment?
Okay. So was it a question about?
On the third one.
Yeah. Okay, so there's a generic problem, namely you can observe something, like you can observe a DNA sequence, and you know that this DNA sequence has some functional meaning, all right? But this functional meaning is not, so to say, rigidly connected to the DNA sequence itself. There's some cloud in sequence space which will lead to the same function, okay? So, you know, when we use handwriting to write a word, right? N different people can write a series of dots on a piece of paper, and they can write the same word, but they, at the level of dots, raw dots on a piece of paper, they will be very different. But you want to compress the information carried by the dots onto the piece of paper into, this was the word hello, let's say, okay? Similarly here, the DNA sequence of this promoter can be compressed into something that has much less variability, namely, what's the level of transcription of the gene that this promoter is controlling, all right? That's one dimension, let's say. And instead of, if I have 100 base pairs with the possibility of four letters in each position, it's a much larger data set. So basically, that's all I mean by data compression. But this is a generic issue in all kinds of, many aspects of biology. It's certainly a big issue in neuroscience, right? Any sensory system takes some kind of input. It might be tactile, it might be visual, it might be auditory, and it converts it into a sequence of spikes on one or many nerves, and experimenters can measure, right? They show something to the retina, and they look at what's coming out of the retina in terms of spikes on a neuron, and you want to be able to read what the spiking neuron is saying in terms of, this is what I saw. I want to be able to redraw the picture that was flashed onto the retina. So this idea that you can have a medium for transmitting information which in principle can transmit much, much, much more information than you're really interested in, and you want to have you want to know how to compress this, in principle, extremely high entropy data stream into something that's much lower entropy and only captures the stuff that you're interested in.
Okay, thank you.
That's what I mean by data compression. So, at one level, I wanted to compress the sequence of the promoter region, 20 bases, let's say, into a binding energy, right? So that's taking something where I could have 4 to the 20 values and turning it into one number, let's say. And so the question is, this is going to come up when I talk about the immune system, this question of how do you, in fact, compress data. And you can't do it a priori, right? There has to be some kind of intelligent modeling. I proposed a certain highly simplified model of how 20 base, the choice of 20 bases, choice of A, C, T, or G at 20 positions was compressed into one binding energy number. So I basically said, let's imagine that it's additive, position by position. That's a guess. It turns out to be a pretty good guess, but it's definitely not 100% accurate. And on the other hand, it does seem in many, many cases to be accurate enough to get a good idea quantitatively of what's going on. So in one of these biological cases, if you want to play this kind of game, in other words, take very complex observations and turn them into something that's telling you something useful functionally, you have to at some point bring in, unless the magic of deep learning or something like that can do this for us without us having to think, it has to be done by thinking and trying.
So, so the energies for the CRP and the RNA polymerase depend on the sequence. And so there are two questions. So does also the energy, interaction energy depend on the sequence? Or is it a hidden variable that introduced? And the second question is this.
So the interaction energy we're talking about, this
epsilon I.
Interaction between the two proteins.
Exactly, yes.
So that doesn't depend on sequence.
Okay, that's just a number. Okay. And then if I understand correctly, the epsilon for the polymerase and for CRP are say linear functions. They are sum of over the residues, right? So is there any information in pair correlations or in pairs? I mean, if you introduce a...
Well, as I was saying, the answer is yes, there is. And it seems to be, at least, frequently it's small enough that you can get away with not having to worry about it. But you have to be very careful. I mean, it's not, or rather, how shall I put it, you have to be modest in your claims.
More questions?
Yeah, so you selected a mutation rate of 12%, I believe. Is there any good reason for that? I mean, probably, so I was saying that in that range, we are incorporating what's happening?
Let's see. 12% was chosen so that there were on average 3 to 4 mutations per binding site, and you didn't want to do too many because you don't want to. There's presumably some basin of attraction in sequence space around the wild type where it still functions a bit. You don't want to have so many mutations that you just completely kick it out of the range of being a functional binding site. That was just it's a choice, and it seemed to be enough to generate enough variation. So it's a matter of something that you make a hopefully informed guess about, and then if it didn't work, you would try a different value. I think we did 25% occasionally, and in a couple of cases, 12% seemed to be enough. If it had been 5%, that probably wouldn't have given quite enough variation. You could have overcome that by getting more data, but at that time, I guess so to say, the long pole in the tent was actually cell sorting. That was rather slow. You couldn't do 1 million cells. Cost too much, took too much time. A couple of 100,000 you could. But again, all of these things change rapidly with time.
So it also, there's more, is there?
Questions about the fluorescent markers. You have 2 different intensities. So you use 3 different proteins or just the different happening?
Try me again. I missed the first part. 3 different...
fluorescent proteins.
It's just good old GFP.
Oh, okay.
No. It's the same GFP in all of the cases. So this plasmid has GFP protein, it's got the modified promoter, it has a antibiotic resistance gene, and various stuff that's going to be useful for the subsequent sequencing and addition. So there have to be various adapters that are coded in. So it's a kind of pain in the neck to construct the plasmid. But the thing that is observed is the fluorescence of GFP, always the very self-same GFP.
Okay. I will try to explain. Actually, right now I'm working with a special transcription factor that has 3 variations: alpha-alpha, beta-beta, and alpha-beta. We know that the energy between the 2 monomers are different, but we don't know which one has a higher probability of binding with the DNA. Is there a way to predict the difference between them, or do I have to just understand it?
A priori, I would say no, but I mean, if you wanted to extract that from an experiment, I think some variant of what I was describing could work. I mean, the larger, so the problem, of course, with all of this is that, let's say, in the case of E. coli, I mean, let's leave aside higher organisms. This has so far all been about bacteria. I don't know, there are several hundred transcription factors. And if you really want to understand the dynamics, you have to have control of some sizable fraction of those. Are we really going to do 200 experiments of this kind? No, we're not. Now, does that mean it's hopeless? I haven't come to closure on that, but the dream was, yeah, we should be able to do a subset, we should be able to do a series of astutely chosen experiments that we could then bootstrap into something that would actually give you a mechanistic view of how gene regulation really works in a real bacterium. And I think there is hope, but it's complicated, all right? And so for the moment, all I can tell you is that if you want to examine particular sub-piece, particular transcription factor and regulatory sequence, yeah, you can do this kind of thing, and it teaches you stuff that's worth knowing. But if you have a high ambition, right, then you have to think about doing this. It's not exactly the Manhattan Project scale. If you really cared, you could do 100 experiments of this kind. But no, the way it's done in biology these days is each one is a separate graduate student who slaves for a year and makes all kinds of mistakes and learns how to do it, and somehow what we're searching for, at least I think we should search for, is ways of, so to say, either managing to do lots of experiments of this kind in parallel so that you can do it in a high throughput way. See, all of this so far has relied very heavily on high throughput sequencing. So that everything is, the data is always one way or another DNA sequence. And so there's another variant of this that I probably won't talk about, where the data is not really DNA, well, it's not regulatory DNA sequence, but it's protein sequence. You can also, there's a whole other story in which proteins talk to proteins, right? That's absolutely key. In fact, that's the key to the immune system, but also in many, many other places in biology. And again, it is the case that, you know, a particular protein talks to another particular protein in a particular sequence, but this generic kind of interaction between a protein of this type and a protein of that type, it's not lock and key. There's a certain degree of flexibility on both sides, and in order to understand how evolution works, you kind of have to understand that flexibility, and experiments of this kind could be adapted to that area, and there have been one or two attempts in that direction. The experiments get more complicated.
Okay, thank you.
Necessary the interaction between the polymerase and the transcription factor. If it's necessary, what is the meaning, the physical meaning of this interaction?
Well, I think at the level of how could it possibly how could the system work? In other words, the transcription factor binding does cause the rate of transcription to go up substantially, right? Really, this is the way it really works. And so mechanistically, from the point of view of equilibrium thermodynamics, that means somehow there has to be an energetic reason for this. And so the obvious zeroth order energetic region is, well, when protein A is close to protein B, when they can actually touch, then there can indeed be an energetic interaction. So that's a thought that people have had for many years, and in fact, the, so to say, previous postdoc in this molecular biology lab of Ted Cox did actually address precisely that issue, and made very, very subtle and sophisticated measurements to, in effect, determine what that interaction energy was. That's why we had a number to compare to, okay? So I think that interaction is necessary. The idea that two proteins, when they touch, you know, when the right parts of two proteins touch, that there can be a strong binding interaction is perfectly straightforward. I mean, that, we know that's true. It happens all the time. Proteins do talk to each other, and they can do so.
The polymerase makes something like an embrace.
It embraces the DNA, so...
I'm agnostic about the exact form of it, right? I had a little tail coming off the polymerase, which flopped over and talked to the transcription factor. The reason why I did that is that the physical sites where the two sit down on the DNA are sufficiently far apart that they're not directly talking to each other. But I believe there is a flexible word, yeah, on the polymerase.
Professor, thank you very much for the lecture, and I was wondering that this kind of binding energy is necessary and an input of for variable sets of parameters, and an input of different models of the whole transcriptome and everything. So, is there any possibility of using this kind of model, or your model in particular, to make predictions about the binding energy of different molecules, or could that be extrapolated? And if not, what's the perspective? What would be needed for us to get that kind of information?
Well, in other words, you're asking, what could I do or what could one do to enhance this to a more comprehensive understanding of how gene regulation is actually working in bacteria? You know, well, yeah, I was bumping up against that question 5 minutes ago, I guess, and the answer is, well, first of all, it's complicated, and I don't have a ready strategy. In other words, you can certainly, you know, you can work, could in principle work in, how shall I put it, steps, right? So there are there are a small number of transcription factors that do a lot, that, you know, regulate many genes at once, broadly acting transcription factors. There are, you know, maybe a dozen of them or something like that in E. coli. So it would be productive, I would imagine, to try to look at them first and figure out how they do their job. How to go beyond that and, you know, find out. So, you know, something like half of the genes in even in E. coli have unknown function, right? Now, you could imagine doing an experiment in which you basically did random variations, not just on one particular promoter, but on, you know, a whole bunch of them, all of them. You could do that. The problem, and I just don't know how to get around this, is that in order to see something happening, you know, you have to change the conditions under which the bacterium is functioning. Or others. So in order to see that, you'd like to know what each of these transcription factors and their associated genes are doing, but you have to see that they do something under one condition, but they don't under another. So I don't know, the question is, how many different conditions are there to worry about? Well, for eukaryotic cells, you know, for human cells, how many cell types are there? Dozens, hundreds, thousands, you know. If you stopped at hundreds, then you would say, well, maybe there are only 100 conditions that, if you really understood how things regulated up and down in 100 different physiological conditions, maybe you would have the wherewithal to start putting together more comprehensive models. I think that, you know, for me, that is the big problem for somebody who wants to think more or less like a physicist about cellular biology. Just how complicated is it? And how is the problem of putting together a useful model combinatorial at the level of hundreds of options that have many different answers, or is it somehow, are there strategies out there that will help you simplify the problem? You know, we need smart people to start thinking along those lines and hoping that life will be kind.
Okay, so let's thank all the speakers of the day, and we come back here tomorrow at 9:30.
Just an announcement, tomorrow the seminar will start at 4:45 instead of 5 o'clock. So all the days the last coffee break will be shorter.